Howdy, folks. Welcome to our fourth week. So, this week we are having our first set of exams. There's exam one and exam two, designed around your lab and your lecture, respectively. So, make sure that you get on that, look at it, take care of it. Don't let it pass you by. It's going to be due on Sunday. And yeah, the questions are pulled from a question bank. So, it's an open book exam. Make sure to just use the materials that have already been presented to you. Do your research, however. I've designed these exams to go and they still are challenging even with access to those resources. So you can't just Google all the answers. You have to actually do some problem solving. So make sure to go and have all your resources ready before you go and throw yourself into it. This week's a very busy week. We actually have two chapters worth of content to cover, as well as the exams. I'm sorry that we're having to go and rush all this, but this is a very content intensive course and it has to meet certain standards. So, given that, I think that we should go and just get started. There's going to be two recordings today. The first recording is this one. The second recording, I assume, is right next to it. So, let's get to it. So, let's pull this up and then this up. Share screen two. Got my little Outer Wilds background, always fun. And from beginning. Here we are. Alrighty. Okay. So this is chapter 19, chromosomes and cellular division. So we have two types of cellular division. All right, so here's the framework for the content for this section. We're going to cover the two types of cellular division, mitosis and meiosis. We're going to talk about the different forms and structure of chromosomes. We're going to talk about the cell cycle and what that means. And then we're going to dive into one of the forms of cellular division, mitosis. We're also going, I'm sorry. It was a two-part outline. We're also going to talk about cytokinesis, which is the essential final stage of cell division, as well as karyotypes and the concept of meiosis, which is the other form of cellular division. So at the beginning, life begins with single cells. It always starts with one cell. In this case, our cell is known as a zygote, and it is formed by the union of an egg and a sperm. So, because it is getting one copy of DNA from the mother and one copy of the DNA from the father, each zygote has double the amount of DNA of a normal egg. Or sorry, double the amount of DNA, one copy from each parent. So we represent this by saying 2N because it is two copies of every type of chromosome. Now, the two types of cell division, meiosis and mitosis, both lead to nuclear division and give rise to offspring. However, in mitosis, we give rise to gamete, also known as reproductive cells, while in mitosis, we create cloned cells, cells that are identical to the parent cell. A multicellular organisms like us use both types of process. So here's a little video that kind of talks about it. Or is this one a video? Huh, thought this one was a video. Oh, yeah, there it is.
Nearly all of the cells in a human are diploid somatic cells that divide by the mitotic cell cycle. The diploid cells of the ovaries and testes, however, undergo a special cell division process called meiosis, which results in the formation of haploid gametes, eggs in females, sperm in males. These two haploid gametes undergo fertilization to form a zygote, which has the diploid number of chromosomes restored. This zygote will undergo the mitotic cell cycle to ultimately produce another adult human.
So as is demonstrated here, we are undergoing when we undergo sexual reproduction, we produce gametes that have one copy of our DNA. Now, that could be any sort of recombination from the parental DNA, but the one copy is going to go and be passed on in the form of sperm in the case of a male or an egg in the case of a female. Now, a haploid cell, a cell that only has 50% of its DNA, cannot fulfill all the functions of a cell. So the egg and the sperm fuse together to get us back up to the diploid number. One cop, one copy from mom, one copy from dad, which in turn allows this complex to go and grow into a full organism, a human. So meiosis is sexual division. That's what we just described. But mitosis is asexual division. So this does not have the reshuffling of genes that we see with sexual division. But so it results in identical bodily cells, and it is the mechanism by which we grow, we heal from injuries, we maintain our bodies. This is a type of cellular division that's done in every form of life, while sexual cellular division, the type meiosis, only occurs in multicellular organisms. Right. And now within meiosis, we give rise to gametes, which have 50% of the amount of chromosomes that you would want. In a female, this occurs in the ovaries, producing eggs. In males, it occurs in the testes and produces sperm. Here you can go and see the processes by which this comes to be and how it transitions from meiosis in the early stage during sexual reproduction and the creation of gametes. And then after fusion, for an event we call fertilization, we end up with a zygote that has a normal amount of DNA, and then it undergoes mitosis for all its non-reproductive functions. So this is normal cellular division. One cell becomes two cells. The daughter cells are identical to the parent cells. Now, chromosomes are tightly coiled structures made out of DNA that are located in the nucleus of a cell. They they are composed of a DNA strand tightly coiled around a specialized protein known as a histone. DNA contains all the genetic information for a cell and also directs body development as well as maintenance. So DNAs are the instructions by which proteins are created. We've already touched on this a little bit, especially with our protein synthesis lab, but DNA is transcribed to a messenger RNA, which travels outside of the nucleus, goes to a ribosome, and then instructs that ribosome on what protein to make. And then proteins go out and they do jobs, they do the actions. So within DNA, so a section of DNA that codes for a particular structure or protein pattern is identified as a gene. So a section of DNA might contain all the information needed to make blonde hair. A section of DNA might contain all the information to go and make white skin. A section of DNA might make all the information to go and make fingernails form. Each one of those are a different gene focused around a different thing. So this is the fingernail genome, and because I have the tiny nail allele, I have tiny fingernails or whatever. So, So somatic cells are the non-reproductive cells of the body. So all cells except for eggs and sperm, the gametes, are considered somatic cells because they have two copies of DNA. They have two pairs of 23 chromosomes, one pair from each parent. They are only capable of mitotic division, not meiotic division. So they can do asexual reproduction, but not sexual. We called the paired chromosomes, the one from your mother and the one from your father, if they are coding for the same genes, they're called a homologous pair. So chromosome one, you get one copy from your mother, and then you get a second copy of chromosome one from your father. So we would call those two chromosome ones a homologous pair. Then, so there are different types of cells that are categorized based off of the type of how much DNA they contain. So a diploid cell has two of each chromosome, 2N, like a somatic cell. A haploid cell only has one copy of the DNA, like a reproductive cell. Genes occur in pairs in diploid cells, and an interesting phenomenon of DNA, and one of the main reasons that it works as a way of conveying information, is that genes are always located on the same positions on the different chromosomes. Like this section may code for blonde hair from my mom, and this section may code for black hair in my dad, but they're both going to be on the hair section of the human genome, which is going to be on a specific location on a specific chromosome. Of the 23 pairs of chromosomes, sex chromosomes are a single pair that determine gender. Interestingly enough, the X chromosome contains a large amount of information that's going to go and be important. That's going to be important for like basic life function. You need your X chromosome to properly form your digestive tract, your cardiac tissue, and a lot of your neural structures. So if you have an individual that's born that was genetically engineered to have YY instead of XY, it would not result in an individual that's more male. It would result in an individual that's missing a lot of organs. The Y chromosome is significantly smaller and codes for significantly less data. So the Y chromosome's main contribution is modifying genes from the X chromosome to express the secondary sexual traits that are associated with maleness. Like in our species, it's sexual dimorphism. We're taller, we have facial hair, have more heavy musculature, external genitalia, things like that. Like if you do not get the Y gene, that you still have all the genes from the X that are still going to go and make a person. But if you remove the X gene, the Y gene does not have enough to sustain you. Just fascinating. Now, we refer to the non-sexual chromosomes as autosomes. They make up 22 of the pairs, while the sex chromosomes make whoa ah while the sex chromosomes are a single pair. Now, every cell is going to undergo a life cycle known as the cell cycle. These are very predictable, pretty standard between different types of cells, and they also tend to stay in those stages for similar ratios of time compared to the rest of their cell cycle. And we can start it at the point when a daughter cell has just been formed from a parent cell. So we have two major phases of the cell cycle, which are interphase and cellular division. So the process of creating the daughter cell begins with mitosis. A single cell will divide into two daughter cells with identical DNA to the parent cell. The parent cell does this by doubling its DNA before division. Here's a chart demonstrating the whole event. So right here, let's say that cell division has finished. We have our daughter cell and it's new. Well, it's going to go and first start in G1. So G1 is the first growth phase. This, it is important to note, is when the cell is functioning to do whatever job it's been assigned. So if it's a skin cell, it's stopping UV radiation. If it's a ciliated stomach cell, it's waving in fluid to go and try to keep your digestive tract moving. If it's a villi in your lung, it's going to go and do gas exchange. After a certain amount of time and a certain amount of growth as a cell, we start synthesis, which is when the DNA replicates itself and doubles. This is also a period of growth. But at this point, more cytoplasm's being made, more organelles are being made, a lot of stuff's happening. Here is where is the final degrees of growth, and the chromosomes start to go and wind up, and cell function starts to gradually decline. In order to go and do jobs and trade in proteins, a cell needs to be able to access and modify pieces of its DNA. It can't do that if it's all tightly wound around a histone, so function gradually falls off. Once we have a double set of DNA and we've pretty much doubled in size, we begin mitotic division, also known as non-sexual cell division. So the first stage of this is prophase, then it goes into metaphase, anaphase, and telophase. And we'll go into greater detail on all of that a little further in. But for now, just know this is the process by which all the organelles in DNA gets evenly divided to either pole of the cell. And then cytokinesis is the process by which the now divided sets of organelles uh, are divided on either side of the pole. And then the equator, the cell starts to pinch and like squeeze itself shut, which in turns separates the two patches into two separate sets, which creates two daughter cells. So interphase is a period of growth and preparation for cell division. It's important to emphasize this is not a rest period. It sometimes is taught as a rest period, but the cell is working during this period. This is when it's actually doing its little cell job. Now, the first one, gap one, is the main focus is on growth. This is before DNA is replicated. This is also when the cell is fulfilling the majority of its primary functions. And the chromosome, we've already talked about that. It's a strand of DNA wrapped around histones. After that, we enter synthesis phase, also known as S phase, which is when the DNA is replicated and then attachments and then anchored at the centromere, which is like it's a guiding spindle for fibers inside side of the cell that are going to direct the majority of this motion. Then we have chromatid. I see. So when the sister chromatids are all right, so during cellular replication, a single chromosome is going to be duplicated because we are doubling all our DNA, which is going to create an artificial system where for a while we're going to be at 4N. We're going to have two copies from dad and two copies from mom, or two copies of whatever ever reshuffling we ended up with. Now, or sorry, two copies from a double set of copies from the parent. So when we have a double set of copies from the parent, growth is we call these chromatids. So they're not sister chromosomes because it's not like one from dad, one from mom, like in sexual reproduction. They are identical in every quality because one is just a replica of the other. After that, we enter G2, also known as gap 2. This is a period after DNA synthesis before mitosis begins. So they have a double set of DNA, but they're still doing their job, and growth is going to continue. Here's how you can go and visit this process. So this is DNA during interphase. The chromatin is not condensed. It's unduplicated. It's just kind of floating around, doing its job. Now, over time, this is going to go and start condensing and replicating. So here it replicates. It hasn't started condensing yet. And then you can see the DNA starting to, during mitosis, wrap itself around these little histone proteins, coiling tighter and tighter, and then folding in on itself until it creates a gigantic spiraled structure. We call this structure the chromosome. Division of the nucleus and cytoplasm consists of two processes. Mitosis is division of the nuclear material in the organelles, and cytokinesis is the final division of the cytoplasm. So this process here, when the walls of the cell, when the cell membrane starts to pinch in a little bit and divide the cell into two components, this is cytokinesis. Here you can go and see what DNA replication occurs, and then during mitosis, these fibers anchored on these spindles start to go and pull apart the DNA to either end of the cell, which in turn creates this division that then allows the cytokinesis to begin without cutting any DNA up or unevenly distributing the components. So division of nucleus and cytoplasm. Uh, So body cells are always dividing. The cells in your body all have slightly different ages, and they're all at slightly different stages of the cell cycle. Right now, millions of cells in my body are currently finishing division or beginning division. Uh, it really just comes down to the fact that we have billions and billions of cells, and so they're all going to be doing slightly different things because each is technically a living component. So the older you get, the less good at dividing your cells are. Part of what causes aging is you basically aren't healing and recovering as fast as you used to because your cells are getting damaged from age and radiation and things like that. So like this is what leads to damage to the liver and the skin in older people. Skin wrinkles are basically the skin not being able to replicate as fast as it's dying anymore. Mitosis occurs in four phases. These are prophase, metaphase, anaphase, and telophase. So let's go through them one at a time. Prophase is the first stage of mitosis. This is when the chromatin forms. Chromosomes are formed as the DNA is wrapping around the histones. The nuclear membrane starts to break down, and then the mitotic spindle near the centromere starts to form. Then we have metaphase, which is where the chromosomes anchor to the mitotic spindles and form up on a line, all the chromosomes together across the equator of the cell. This makes sure that the cells are evenly distributed and also ensures that each daughter cell will get one chromatin from each of the 46 chromosomes. You're not going to end up getting like three copies of one, two copies of two, eight copies of three. You're going to go and get exactly one chromatid from each of the 46 chromosomes. Then we have anaphase. I remember this one because Anna's the sister in Frozen that like freaks out and gets separated from the other sister. So sister chromatids begin to separate. They split at the centromere. And then once separated, they are considered individual chromosomes, even though they haven't really changed much other than being broken off from the sister chromatid. The next stage is telophase, and this is when the nuclear envelope reforms around each group of chromosomes at the pole, and then the chromosomes start to decondense, and the mitotic spindle gets absorbed for nutrients, and the cell resumes its function, or begins its function, depending. So let's go and watch this video. This is just whenever I like to use multimedia approach to education, and I feel like if I explain something poorly, but I make sure that you also get another one to two to three resources on that subject. Someone is going to go and be able to explain it right. So for this one, let's do this video on mitotic division.
A cell is ready to enter the M phase of the cell cycle. Chromosomes have already been duplicated, and each consists of two identical sister chromatids. Chromatid is one of the newly replicated DNA double helices, plus its associated proteins. In an interphase cell, there exists, just outside the nucleus, the centrosome, a cellular structure that acts as an organizing center for the assembly of microtubules. Mitosis begins with the events of prophase. In the cytoplasm, prophase is marked by the formation of the mitotic spindle, assemblies of microtubules emanating from each centrosome. The chromosomes condense into compact structures that can be seen with a light microscope. Before the cell enters metaphase, the nuclear envelope breaks down. After the nuclear envelope is disintegrated, spindle fibers from each pole of the mitotic spindle attach to one of the two sister chromatids of each chromosome. A tug-of-war is occurring, with spindle fibers pulling each chromosome in opposite directions. In the next stage of mitosis, metaphase, the chromosomes complete their journey to the middle of the cell. Each chromatid now faces the pole opposite that of its sister chromatid. At the start of anaphase, the link joining each pair of sister chromatids is broken. The spindle fibers shorten, pulling the chromosomes to opposite poles. This segregation of sister chromatids to opposite poles ensures that each daughter cell receives the same complement of chromosomes. In telophase, the final stage of mitosis, the mitotic spindle disintegrates, and a nuclear envelope starts to form around each set of chromosomes as they return to their original uncondensed state. Once two independent nuclei have formed, mitosis is complete. Meanwhile, a cleavage furrow has begun to form, and immediately after the end of mitosis, the cytoplasm of the cell divides in a process called cytokinesis, forming two daughter cells.
Alrighty, so that's just what I said, but explained with visuals and movement. I think that's a useful tool. So here's how this actually physically looks at the different stages under a microscope. So all these are cells that have been treated with a dye that binds to nuclear material. And so you can see the kind of dark bundles that are the genetic material inside of the cell. You can actually see the individual chromosomes on this one lined up across the equator. It's pretty cool. Now, if you look at this one, then we go through anaphase. It's very distinct in anaphase and telophase. And then finally, as we finish our cytokinesis and re-enter uh, interphase, the chromosomes unwind and decondense inside of the nuclear envelopes. Now, cytokinesis is the division of the cytoplasm that begins during telophase. This is interesting because it involves rings of microfilaments and rings of actin and myosin. So, actin and myosin are the same fibers that control the contraction of your muscles. So, anytime you go and like pop a gun or go and try to show off for the ladies, what you're doing is you are using actin and myosin in the same way that cells are being divided. It's a cinch mechanism. One of the proteins can travel across the surface of another protein, and so it pretty much just pinches the cells apart. So occasionally, when trying to go and evaluate individuals for genetic diseases, a very useful tool is known as a karyotype. So they use radiation to take a scan of a chromosome. They arrange the chromosomes to match them up based off of size and centromere location. And then that lets you examine them for abnormalities because every human should have the same number of chromosomes and every human should have the same ratio of chromosomes. So we can work off of here. This is a standard male karyotype. Here you can go and see we have 1 through 23. These are our somatic chromosomes, our non-sexual chromosomes. And then here, or sorry, 1 through 22. And then here, 23, we have the sex chromosomes. So here we have an X and here we have a Y. And you can see because the Y codes for so much less information, it's significantly smaller. It's a modifier. It does not contain any endemic structures. So gametes, the sex cells, refer to egg or sperm. They're haploid, meaning they only have one copy of DNA. They do not have a double copy. And they have one member of each homologous pair of chromosomes. So they do have one copy of each type of DNA, but they only have one copy instead of the standard two. So this is formed through meiosis, which is basically mitosis so nice we did it twice. This is what happens when a cell undergoes DNA replication and then divides and then immediately divides again without undergoing another stage of replication, resulting in 4 haploid daughter cells. So 4 daughter cells that have half the DNA of the original parent. Meiosis is designed to keep the number of chromosomes constant from generation to generation and allow for genetic reshuffling or allelic reshuffling in order to create increased genetic variability in a population. So, if I have a species that explodes when it touches water, it's going to die in the next rainstorm. If I have a species where half of the members explode when it explodes when they touch water, half the species is going to survive the next storm event. Genetic variability gives more resiliency to a population because it leads to more novel solutions to problems. In general, when you're trying to solve problems, the more solutions you have that you can then sort through and process to find the good ones, the better. You want as many options as possible, right? So meiosis involves two cell divisions, which are meiosis 1, which separates the homologous chromosomes, and then meiosis 2, which separates sister chromatids. So we basically duplicate the DNA, divide it down into the cloned chromatids, and then separate those. Each division has its own prophase, metaphase, anaphase, and telophase. So again, it's mitosis so nice, we do it twice. This is this kind of visualized. So here you see a diploid parent cell containing two chromosomes, and each one of them is part of a pair of homologous chromosomes. We have it has under this normal cell has undergone DNA replication, and now it has a double set of each chromosome. Oh, I'm sorry. DNA replication occurs, double set of each chromosome. Then we have meiosis 1, where they are divided. And other than the joining with the sister chromatids, this is going to be pretty similar to the diploid parent at this stage. This is almost a replica of the parent, but not quite because of a couple different phenomenons of how the chromosomes interface with each other. So meiosis 1 is pretty much the same as mitosis. The thing is, we then begin meiosis 2, which is a second stage of division. That leads to four daughter cells with 50% of the DNA of the original parent before cellular replication or DNA replication. So here, meiosis 1 is preceded by interphase. Each chromosome consists of two attached chromatids. We have reduced division, which produces two haploid cells, and each daughter cell has 23 chromosomes, one member of each homologous pair. So we haven't lost any genes. We just have reshuffled and lost some alleles within a gene. Prophase is the process by which the chromosomes condense, the mitotic spindles form, and the nuclear membranes break down. Then we have synapsis, which is when the homologous chromosomes pair. And then at that point, we engage in something we don't see in asexual reproduction, known as crossing over. So crossing over is at a certain stage of meiosis 1, random chunks of genes are going to be arbitrarily switched between two sister chromatids. So we're still changing out the inputs for the same gene. Like we're switching out the black color gene with the white color gene for hair color. So the same gene is there, but the alleles are being reshuffled. This is another point onto why sexual reproduction leads to way more genetic variability than we see in non-sexual reproduction. Metaphase 1, homologous pairs line up the midline. We get two lines of 23, and then the homologous pairs attach to spindle fibers. So because they're lined up randomly and because of crossing over, we call this independent assortment. It's basically the effect of randomness upon our genetic frequencies, our allele frequencies. In small systems, that can actually lead to pretty large skews, and we tend to go and refer to that as a genetic drift. Then you have anaphase 1. Homologous pairs of chromosomes separate and move to opposite ends of the cell, and now each homolog still consists of two sister chromatids. And then finally, we enter telophase, which leads to cytokinesis, and then that brings us on to interkinesis, which is like a very brief interphase-like period before mitosis 2 starts. There is no DNA replication during this phase. Like we have finished all our DNA replication. These are two, in theory, normal cells that are just going to divide again very soon. After that, we enter the second stage, prophase 2, same things happen. Metaphase 2, same things happen. All the chromosomes lined up along the equator. Anaphase 2, everything gets dragged to opposite poles, split in two. And then telophase 2, and now we have four haploid daughter cells. It's just the same thing happening over and over again in different patterns. So important features of meiosis 2 is the separation of the two sister chromatins of each chromosome. So this video can kind of explain the process I just described. but with, again, a flowchart of visuals. We also have slides that you guys can use to study that cover all these stages as well.
A cell that undergoes mitosis produces daughter cells that are dramatically different from the daughter cells produced by a cell that undergoes meiosis. Let's compare these two processes, beginning with a look at mitosis and meiosis 1. In both mitosis and meiosis 1, cells begin with replicated chromosomes, each cell having two chromatids per chromosome. In meiosis 1, the homologous chromosomes pair with each other and come to the middle of the cell together. In mitosis, the homologs move independently of each other. In mitosis, the chromatids separate. In meiosis 1, the homologs separate. In mitosis, a diploid cell divides to produce genetically identical diploid cells. Each cell contains two complete sets of chromosomes. In meiosis 1, a diploid cell divides to produce genetically unique haploid cells. Each daughter cell now contains just one set of chromosomes. Meiosis 1 is known as a reduction division because the outcome is a reduction of chromosome number.
Sorry about that.
Here are the processes, beginning with a look at mitosis and meiosis 1. In both mitosis and meiosis 1, cells begin with replicated chromosomes, each cell having two chromatids per chromosome. In meiosis 1, the homologous chromosomes pair with each other and come to the middle of the cell together. In mitosis, the homologs move independently of each other. In mitosis, the chromatids separate. In meiosis 1, the homologs separate. In mitosis, a diploid cell divides to produce genetically identical diploid cells. Each cell contains two complete sets of chromosomes. In meiosis 1, a diploid cell divides to produce genetically unique haploid cells. Each daughter cell now contains just one set of chromosomes. Meiosis 1 is known as a reduction division because the outcome is a reduction of chromosome number. These two cells are daughter cells produced from meiosis 1. Let's use just one of them to compare meiosis 2 to mitosis. A cell entering mitosis is diploid, with two complete sets of chromosomes. A cell entering meiosis 2 is haploid, with just one complete set of chromosomes. In both cells, each chromosome has two chromatids. In both mitosis and meiosis 2, the chromosomes migrate to the middle of the cell. Then, the sister chromatids of each chromosome separate to form independent chromosomes. In mitosis, a diploid cell divides to produce genetically identical diploid cells, each with two complete sets of chromosomes. In meiosis 2, a haploid cell divides to produce genetically unique haploid cells, each with one complete set of chromosomes. If we consider that meiosis 2 actually began with two cells, then the result is 4 genetically unique haploid cells. In both mitosis and meiosis 2, the division is not a reduction division. Only in meiosis 1 are the number of chromosome sets divided by half.
Okay, yeah, so that's just unifying the concepts and putting them onto a model. I really enjoy that aspect of science. I call it the emergent properties. So here is just a chart to guide you through meiosis 1. This is a useful study resource. I would suggest you utilize it. Here is the same chart, but for meiosis 2. And here's a visual on how mitosis and meiosis are the same and different. So following meiosis, changes in shape and function occur, which result in functional gametes. So when we are creating sperm cells, we call this process spermatogenesis. These sperm cells are highly specialized for transporting genetic material. They have flagellum, they go towards certain chemical signals, they have energy stores, and four are the final product. oogenesis, also known as egg creation, produce up to three polar bodies, which are non-functional, but only one oval. So when we are specializing into creating sperm cells, every single one of the spermatids is viable. So each of the daughter cells will become a living adult sperm cell. When we are making an egg, the cytoplasm is not evenly distributed during division. And so we end up just creating one gigantic daughter cell that contains most of the cytoplasm, and then these little polar bodies that are absorbed for nutrients. So egg formation is more efficient when we are, so, or sorry, meiosis is more efficient through sperm formation than it is through egg formation, because egg formation is about accumulating and maintaining calories. Sperm formation is about spending a minimal amount of calories to send out as many messages as possible. Different biological objectives that relate to different life strategies. So during meiosis, genetic variation is created through many mechanisms. We have crossing over, which is the exchange of DNA between maternal and paternal homologs. So gene one in dad is going to be trading chunks with gene one in mom. This happens during synapsis and prophase one. Then we have independent assortment. Maternal and paternal homologs lined up independently, respectful to the poles. So that means that after the initial reshuffling, it is then arbitrarily decided which copy, the maternal or the paternal copy, is going to which daughter cell. So this also leads to allelic reshuffling. This is how crossing over works. It is just they pretty much just touch each other and chunks jump from one to the other. So two equally possible ways that homologs could align during metaphase one. So this is just kind of demonstrate that this is random. There is independent assortment. So on this lineage, we end up with pink and blue. On this lineage, we end up with just blue and just pink, despite the fact that we start with the same chromosomes, because the chromosomes are independently arranged arbitrarily. It's random. Here's a list just kind of comparing the differences between the two groups. Mitosis and meiosis, one is cell, and just through it all. It's a summation of what we just did. Occasionally, issues occur. So non-disjunction is a phenomenon by which the chromosomes are distributed improperly during meiosis 1 or 2. This leads to abnormalities in development and miscarriages. This is also the process by which a lot of the missing chromosome or extra chromosome genetic diseases come about. So here you can see abnormal disjunction in the first division, followed by normal during meiosis 1, and the normal division during meiosis 2 leads to two chromosomes that have an additional or two daughter cells that have an additional chromosome and two daughter cells that have no genetic info at all. So if this was an oocyte and the rest of these were polar cells, then this would not be a viable division. If all of these were sperm cells, these two would probably not be viable, except in the case of very few genetic diseases like Down syndrome. In this division, the first division's normal, and then the second division is abnormal. That leads to missing chromosomes, extra chromosomes, and then one chance at a standard division, a successful division. So we give these different names depending on how the chromosomes are distributed. So the first one is trisomy, which is where there are three copies of a chromosome in a cell. So instead of having one from mom and one from dad, you might end up with both from dad and one from mom. And then there's also monosomy, which is where only one representative of a chromosome is present. So instead of having two copies, you'll just have one. Here are a couple common diseases. So Down syndrome, that's where an infant is born with three copies, a trisomy of chromosome 21. It becomes more the risk for trisomies and errors in division does increase with age. However, uh, as time has gone on, this has become less of an issue. I'd honestly say there's not really an issue with trying to start a family when you're like 40 or 45 at this point because of how good we are at dealing with this particular set of conditions. Down syndrome is one of the few non-lethal ones, but it does render the individual sterile so that they cannot pass on their lineage because you need to have a number of chromosomes that can be evenly divided in order to create uh, successful gametes. They don't have the right number of chromosomes to do this. They can't produce gametes. Non-disjunction can also occur with the sex chromosomes. However, this tends not to be lethal as long as the individual receives at least one copy of an X chromosome. So when it happens during sperm formation, you either end up with sperm that has no DNA, it has no sex chromosome, or sperm that has a double X, or a sperm that has an X and a Y instead of just a Y, or an X. So non so during egg formation, you can end up with an egg that doesn't have any sex chromosomes. This is almost always non-viable because there's no if they are lucky, they get an X chromosome from their father. If they are unlucky, they get a Y and then they do not come to term. There are a couple different names for different distributions of sex hormones, and this is how disjunctment occurs with the sex chromosomes. So here you see it didn't divide right during meiosis one. This one got a double of X. This didn't get any X. And so when this then is by an X or a Y, if it's a Y, it's inviable because we don't have any of those organ formation chromosomes. If it's an X, we end up with something called female Turner syndrome, which is where you only have one copy of an X. This leads to not express because of the lack of an X and the lack of a Y to go and cover up with other traits. This usually leads to individuals that are sterile, often very petite, often sub-average intelligence, and often pretty extreme skeletal deformities. If you end up with a double X and a Y, you have what is known as Klinefelter syndrome. And if you end up with 3X, you will present as a female with 3X. Now, this will affect the fertility of this individual because they are much more prone to non-disjunction when they're producing eggs, but they can produce healthy, viable eggs and also tend to not present with any negative physiological effects. Klinefelter syndrome, it leads to sterility, reduced expression of male secondary sexual characteristics, and occasionally expression of female sexual characteristics. You will occasionally find individuals who have, because of this hormone deluge that they're being subjected to, they might have half of the reproductive organs of each group neither functional contained in the body, and they might not figure this out until they go in and be like, hey, how come I can't have kids? And then they figure out, oh shit, there's a random ovary tucked behind my kidney, stuff like that. So given that, here were our learning goals. You should be able to compare the role of meiosis and mitosis in the cell cycle, differentiate between chromosomes and genes, autosomes and sex chromosomes, and diploid and haploid cells. And you should also be able to draw the cell cycle, label each phase of mitosis and interphase, and describe each event and the significance of each phase. Uh, and then I think it's worth understanding what a karyotype is, being able to define cytokinesis, and being able to list all the phases through interphase up to telophase of mitosis and meiosis. And you need to be able to discuss what that means. Alrighty, folks. So let me just get this closed. There we go. All right, folks. So that is our first presentation for the week. This information is going to be included on the exam, which is due online on Sunday. There are two exams. You have a lab exam and a lecture exam. The material from this week, including these two videos, are included on that. So it is worth reviewing these videos. With that, I also included a couple additional study resources for this week. I found a couple copies of a study guide for someone else's presentation, but that someone else actually designed this course originally. It's been modified a couple times since then, but the content's still really good, and I like them as study guides. I'd suggest you look at them. In addition to that, I also included our normal study guides. So you have a double set of study guides. All righty. With that, I hope you guys have a wonderful time, a wonderful day, and good luck on your exams.